Hello everyone. Today I am going to discuss about a very important technique in biochemistry, that is sonication. So sonication, first of all, let's go to definition of sonication. Sonication is act of applying sound energy to agitate the particles, and it can be used for various purposes such as extraction of multiple compounds from plant, microalgae and seaweeds. So when you will look into the instrument sonicator, it is basically consists of three parts. First is generator, second is transducer, and the third part is sample. So generator is used for transforming the input electrical power into electrical signal that drives the transducer, and जो transducer है that will convert electrical signal into vibration or ultrasonication waves, and that wave will go to the sample and that will lead to cavitation or disruption of the sample particles. On the basis of method, sonication can be of two types. First is direct sonication method, second is indirect sonication method. In case of direct sonication method, as it is visible in the diagram, probe is dipped directly into the sample or probe is directly inserted into the sample, and it is most common type of sonication which is used in any biochemical laboratory. In this method, energy is transmitted from probe to the sample directly because probe is in direct touch with the sample. This is high intensity process, and in this case, as energies, ultrasonic waves are directly passing from probe to the sample, so sample processing or sample destruction take place very easily in very less time in this particular technique. The volume of the sample which can be taken for sonication, that is taken according to the diameter of the probe tip. So probe tips are made from the titanium, which is non-reactive metal, so that your probe does not react with your sample. Second type of sonication is indirect sonication method. In case of indirect sonication method, there is no direct contact between your sample and probe. Samples are put into a water bath and probe is dipped into the water bath, and ultrasonic waves which are generated from the probe, they travel through the water to the sample. So energy is transmitted from horn to different tubes of the sample. So this model is the preferable model when you have multiple sample and sample volume is very low. Next, uses of sonication. Sonication process has large variety of applications in biochemical labs. It can be used for disruption of cell membrane. It can be used for cleaning purposes also. Let's go through some of these applications. As it is written in the diagram, in the slide, sonication mechanism is used for cleaning purpose, which include cleaning of the particle which are adhered to the surfaces. It can, in the laboratories, it can be used for cleaning of spectacles and jewelry also. Artificial aging of the liquors like alcoholic beverages is done with the help of sonication. In case of food industry, sonication is playing very important role. It can be used for dispersion of emulgators, and it is also used for speeding up the filtration process in food industries. Different parts of soils can be studied with the help of sonication techniques. So this diagram is showing you two types of sonicator, which is probe sonicator and bath sonicator. So जो probe sonicator है, that is type of direct sonication method, and bath sonicator is type of indirect sonication method. So in this case, as it is clearly visible, probe is in direct contact with the sample. In case of bath sample, probe is in contact with water which is present in the water bath, and samples are put into the water bath, and ultrasonic waves travel from probe via water to the samples. Next slide is showing you the differences between probe and bath sonicator. First of all, definition. Probe sonicator is the equipment which is used for direct sonication, in which probe is directly dipped into the sample. In case of bath sonicator, it is indirect sonication method, and probe is dipped into a water bath, not directly into the sample. So this is probe sonication is direct method, bath sonicator is indirect method, as I have already told you. In case of contact with sample, probe sonicator, probe is direct contact with the sample. In case of bath sonicator, probe is in contact with water bath. Energy input is high in case of probe sonicator, and it is low in case of bath sonicator. Probe sonicator is suitable for small, not that much suitable for small sample. It is suitable for large samples, and bath sonicator is suitable for small samples. There are very high chances of cross contamination in case of probe sonicator, because probe is in direct contact with sample, and the chances of cross contamination in case of bath sonicator are very less. When we will go to number of samples which can be processed with the help of sonication method, probe sonicator, with the help of probe sonicator, one sample can be processed at a time. In case of bath sonicator, multiple samples can be processed at a time. So this is all for sonication. Thank you for your kind attention.